Hello, my name is Fran and I'm a member of the Alzheimer's Research UK Thames Valley Network. I work as a scientist here at the ARUK Oxford Drug Discovery Institute, for short the ODDI, aiming to defeat dementia. I'm sitting outside the NDM research building, which accommodates the drug discovery's facilities. But let's go inside and see what's going on in there. We will go all the way up to the top floor. The ODDI is one of 3 ARUK-funded institutions. And our group here in Oxford is a team of experts in different fields such as chemistry and biology, combining both academic and industry knowledge. Together, we want to tackle Alzheimer's disease along with other forms of neurodegenerative diseases and discover novel, effective therapeutics. We are sharing a large office and lab space with other groups, which enables us to communicate and discuss our science and help each other. I will now introduce you to some of our scientists that work on ARUK-funded dementia research. All of us are using and sharing a wide range of equipment in our lab. We have all been working throughout the pandemic under safety measures to ensure our research progresses. The journey of drug discovery here in the ODDI starts in the chemistry lab, where compounds are made from tiny molecules. Personally, it always reminds me of cooking, mixing ingredients that react together and form an end product. I want to introduce you to one of our hardworking chemists, Trifon.
Hello, my name is Trifon and I work in drug discovery. I am making a compound that aims to prevent processes in brain cells that lead to the development of Alzheimer's disease. The compound stops the cells from producing an unwanted complex of proteins that, if left alone, would lead to Alzheimer's. I make the compounds by combining different reagents we have in the lab. Then I purify the final product to test in our experiments.
So we have the compounds made by the chemist, and they prepare them for us in a concentrated stock solution. But what happens now? Please meet my colleague James to learn more about how we use these stock solutions of these compounds.
Hi, my name is James. I'm a biochemist and I am testing new compounds sent to us by the chemistry team to see if they alter the activity of our target enzymes. A target enzyme is one that we have identified as playing a role in causing dementia, and we hope by altering its activity inside the body we can help treat the disease. When we receive the chemicals, we dispense them into these plastic plates with tiny wells for each chemical. To dispense the chemicals for testing, we use this machine here called an echo. This machine takes our highly concentrated stocks of compounds and uses sound waves to ping tiny droplets of drug from our source plate into the wells of our experimental plates. The robot arm attached to the echo can be programmed to automatically insert and remove the plates. This allows us to precisely dispense thousands of compounds much more quickly and accurately than we could do by hand. The experimental plates are then ready for the enzyme to be added or can be stored in environmentally controlled pods until we need them.
Please meet Grace. Grace is not only committed to ARUK-funded science, but she is also a member of the ARUK fundraising team here in Oxford, helping to support dementia research. Grace will tell you more about the effects of compounds in organisms, more particular in cells, the smallest functional units an organism is made of.
Hello, my name is Grace and I'm a research assistant here at the ODDI. Currently, I'm researching the role of inflammation in the brain and how this can be prevented with the new drugs made by medicinal chemists like Trifon. To look at the strength of our new compounds, we first do this in cells that are grown in a flask. Here you can see the layer of cells growing under the microscope. From the flask, we transfer the cells into a plate and add each new compound to different wells along with a stain. This makes the cells fluoresce brightly, so we can see different parts of the cells clearly when we image them. And now for the fun part. This is our Opera Phoenix. It is a specialized piece of equipment that allows us to take images of the cells in great detail and see the effects the drugs are having. For example, using the staining, we can see the appearance of these bright specks inside the cells, which are signs of inflammation. This one is important because it doesn't have any specks. Using this technique, we can look for new drugs that prevent the appearance of these specks. The aim is that these can be taken further as new drugs for Alzheimer's disease.
And now it's my turn to tell you about my research. This equipment, called an octet, is used by myself a lot. I'm investigating a protein that is involved in the progression of Parkinson's disease. Ideally, we want to inhibit this protein with inhibitor compounds to stop the disease progression. I'm using these sensors that will be coated with this protein, and these sensors will be dipped into inhibitor compounds. With this technique, we can optically detect interactions between target and compounds. We can measure how well these compounds bind to our target, which is a very important information concerning the effect of this compound as a potential treatment for patients. Hector is working in the darkroom lab, where light-sensitive experiments are taking place.
Hi, my name is Hector, and I'm a research assistant here at the ODDI. Today, I'm using our Flipper Tetra instrument to evaluate the effects of some of our new compounds on a protein called SHIP1, which is involved in the progression of Alzheimer's disease. We are trying to find a drug that inhibits SHIP1 to hopefully help patients. With this machine, which is basically a very precise camera, we are able to detect phosphate as it is produced by SHIP1 and ultimately find compounds that inhibit its activity. So I'm actually looking at these two curves here. This one represents the reactions as it happens without an inhibitor, and this one represents the reaction with an inhibitor. So we're looking at compounds where the reaction will look like this rather than like this. So this is the undisturbed reaction, and this is the one when we've added our compound, and we're looking basically for that reduction in signal. So here on the right, I got basically all the wells in the plate, and then I can see all the different curves. Click on them and get the different curves that come out, so I can evaluate each of these represents a specific compound, and I can look, oh, this one didn't inhibit or another one inhibited basically. This one, reduction. This one a bit less of a reduction. And I'm looking here, there's a difference. These ones are active, this one's inactive. You can see here a trace which represents the amount of phosphate produced and how one of the compounds stops that reaction.
Joining us in the darkroom is Juliane. She's working with a type of brain cells called neurons. These can be grown in a dish. These neurons are very important for studying and understanding the disease progression of dementia.
Hi, I'm Juliane. I am using the microelectrode array, or MEA, to study the function of neurons in vitro. Neurons are the cells in the brain which commonly die in neurodegenerative diseases, such as Alzheimer's disease. Under normal conditions, neurons form spontaneous networks and communicate with each other by generating electrical signals. And with the MEA, we can measure this electrical activity in real time. Therefore, the neurons are cultured in a specific plate, which has a grid of small electrodes embedded in the culture surface to detect the electrical activity. This plate-wide activity map shows the neuronal activity across the plate, with each square representing a different well. If we take a closer look at the specific well, we can observe the neuronal activity in the form of so-called spikes and network bursts, which are key features of healthy and mature neuronal networks. This is a great system to characterize our neuronal cultures and to study disease-related mechanisms. We also use the system to test compounds in order to see whether they modulate neuronal activity and to screen compounds for safety and efficacy.
Special thanks to Chris. Without his coordination and help, the tour today would not have been possible. I hope this virtual lab tour gave you an impression of what our ARUK-funded scientists do and how the funding is being used to help to defeat dementia. We had a lot of fun making this video for you whilst showing our science and equipment. We are very proud to be part of ARUK-funded drug discovery.